So I value this particular lecture even more than the previous one. Okay? Get the important concepts, get inspired, get the curiosity all the time. When you do research, okay, give a reasonable doubt, and then you can start your hypothesis. Okay, so here the cartoon says, with the scientist A tell the scientist B, say this is the Hitler's brain. And then you say, wait, how do I know this is the reality? This is really Hitler's brain. You could ask him, show me some evidence to prove this is Hitler's brain. Right? So you need to have certain reasonable doubt. Someone tell you this drug is useful. You ask them, give me some evidence to convince me. Right? Otherwise, it's not science. In the English term, it's called snake oil. Okay? Someone selling the snake oil say this could cure 1,000 kind of different disease. But what's inside the snake oil could be able to do the job? You don't know. But indeed, maybe the snake oil is really not effective. Right? But you need to initiate your hypothesis. Okay, some component in the snake oil I could purify, get a component out, get the formula, and I do the experiment to prove it is effective in heavy cancer. Then that's good. You initiate your hypothesis with a reasonable doubt. Okay, I don't believe there's snake oil now, but if I could do an experiment to prove some component in the snake oil could be useful. Okay, and how do you do your hypothesis? You could do all the things, design experiments to prove your hypothesis is correct. Very important. And we talk about hypothesis driven. Okay? Here, the cartoon show here. Maybe the dinosaurs disappear because they smoke too much. Right? Is it possible? That could be dumbest idea, right? Dumbest hypothesis. But it's still useful. Even this hypothesis could be wrong. But you start doing the first step. I give you the number one hypothesis, okay, smoking. Yeah, but at that time, how dinosaurs know how to smoke, but you never know. Right? They may be not using the cigarette like we are, but they probably particularly go to some volcano area, pick up the branches, and start inhaling. How do we know? But even not a dumb hypothesis is still useful. Okay, I just encourage you to think. Okay. Of course, we know now smoking is bad in many countries. We already proved it. Epidemiology, all the chemical compounds in the smoke in the cigarette show that many of them are carcinogens. One particular kind of carcinogen may be harmful for certain type of disease. We don't know because it's smoking, not only bad for lung cancer, as we know. Smoking is bad for other type of cancer, including bladder cancer, including other type of cancer. But why? You ask why? Why smoking is bad for bladder cancer? Our brother smoke. Some of the carcinogens be circulated into that particular area, make them vulnerable. And you could initiate a hypothesis to prove it. When you do experiment, you need to keep a good notebook. I want to ask, for those of you who keep a good notebook, even in the class, raise your hand. I see about 15 also. For those other people who cannot keep a good notebook, you should keep on trying. It's very important. not only in class, but also when you're doing experiment. Okay? Keep such a good notebook is very important when you become a scientist. The reason why, to reduce error, to make your observation repeatable. Okay, here the cartoon show one thing. The guy find a HeLa cell. Anybody know HeLa cell? What is HeLa cell? Raise your hand. Okay, 1, 2, 3. Maybe you could tell. What's a HeLa cell? What's a HeLa cell?
It's a cell come from a woman cervical cancer, like so the cancer cell that was like taken out and it's period, like we would call it like cancer cell.
Good. Okay. HeLa cell, many of you should know because this is probably the first well-established human cell culture. Okay. And the HeLa cell, like he said, he gave a good, correct answer. It's coming from cervical carcinoma, from a woman, okay? The point. Everything was going along fine until they discovered their HeLa cell line. Y chromosome. Why Y chromosome in HeLa cell? Everybody get a joke? HeLa cell is a female cervical carcinoma, right? Every cell, you have a 23 pair. And there is no Y chromosome in this HeLa cell, supposedly. Because the female, only XX chromosome, no XY. So when you do experiment, be very, very careful. You need to keep a good look. I think the situation here is because the guy maybe got his cell contaminated. Okay? Maybe contaminated with the prostate cancer cell. Okay? So be alert, be careful, be mindful. Okay? It's very important you have the concept. Keep a good notebook. The cell I got from, maybe it's not your fault. You get this cell from the other guy. The other guy told you, this is a HeLa cell, go use it. And you take it for granted. You say, oh, this is a HeLa cell, you use it. Until you do cytogenetic, you find out, wow, this cell has a Y chromosome. How could that be? Okay? But you keep a good record because you know this is cell line you get from that person. Then you know maybe this was something mistake happened. Okay? So keep a good notebook. It's so important so you won't waste a lot of your time. Okay? You're doing the experiment again and again. You need to use this HeLa cell, but how come it doesn't behave like HeLa cell? I tried to do an IP Western to see the E6AP and the HeLa cell, and they come out with the PBP3. I couldn't see it. What's wrong with it? You keep a good note. You know the HeLa cell from other people. And you go to ask him, where did you get your HeLa cell from? The guy may say, I get it from other people again. But at that time, you could make a judgment. Maybe if I do continue to do the IP Western, couldn't see it, you need to change route. And get another batch, HeLa cell from other people to see if I can see it. Okay? Keep a good notebook, it's very important. Okay? I think the cartoon here shows something. Okay? I just want to let you know it's very important. So, the other thing is when you do experiment, think simple, okay? But it open for much more complicated observation. Okay? We don't want to have a very complicated thinking at the beginning because that will make you confused at the beginning. You don't need to do that. You do one by one. Okay? Here, just show a very simple genetic, right? The guy married this person. I'm going to have a baby like this with a beautiful, maybe a mermaid-like daughter. And see, he was thinking about the handsome boy with the stallion-like the characteristic. But in the end, they come out something like this, unexpected. Right? True. When you design hypothesis, you design your experiment, you only think about, okay, A plus B, I'm going to get something. But if it come out with some other things, be prepared for it. If only it's not an artifact. Right? Be prepared for it. It usually happens now. And you will have a novel discovery. Think simple. That's okay. Okay? Just like this guy, just like this lady. Think simply. It's okay. Very important to have a simple concept. But really prepare for the complicated situation. And it's always happened. Because when we do this experiment, we think that this way, along this path it will happen. Sometimes come up with the other. You just need to be open for that mind. Because if you think that you have simple-minded and then say, oh, forget it, I didn't see the observation, and you give it up, sometimes you give up the very novel discovery. Think simple, open for the complicated discovery, observation. When we do experiment, we do one step by one step. A very important concept is that it's like a window opening process. Or it's like the blind man touch an elephant. Okay? You all know the story, right? The blind man touched one part of the elephant, and then the guy touched the body, thinking the elephant is like a wall, okay? And then the guy touched the leg, I think it's like a color. Touched the tail, think of the elephant is like a snail. They're all correct. Okay? So when you do experiment, you do one way, you have this observation, you say you do one, you try to see protein A interact with B, and you find that protein A interact with B. Correct, that's correct. The other group of scientists, they find protein A interact with C. It's also correct. You cannot say, I find protein interact with B, but you find protein A interact with C, that's wrong. You are everybody doing single step to make the whole picture complete. Okay? And I like to make an analogy, say the window opening is the same situation. There's a house full of windows right next to the beach. In the front, there's an ocean. In the back, there's a mountain. The first guy coming, open the window, face the ocean, say, oh, he make a conclusion. This house face the ocean. He's correct. The other guy coming, open the window, see the mountain, they say, oh, this house, there's some structure facing the mountain. It's correct too. But until we open all the windows, we find out this house sit right on the beach with a mountain in the back. Okay? I just want you to know that every group of scientists makes certain contribution. When we have thoroughly investigated every possibility, we are to see the whole picture. Okay? And everyone is doing part of it. You're opening one panel window, he's opening the other panel window. You are touching one part of the elephant, he is touching another part of the elephant. Is that concept, okay? Next one I will share with you, as I told you the cartoon there. It's a tough process, long process. You need to be patient. There's no royal road, as we know. There's no royal road for learning, for being a scientist. It takes such a long time to cultivate a scientist. But it all starts from you now, and you need to do it now. Cultivate. The cartoon show you want the Captain Hook had the parrot, right? The boy come on and say, Captain Hook, I want a parrot like you. Captain give you an egg. The egg needs to be hatched to become a parrot. You need to be patient, okay? But do it persistently. It is such a long road, long process, but you need to be patient. You need to be persistent, okay? We know to build up science research, okay, not only difficult in certain countries, even still difficult in America. You would think America has much better opportunity to become a scientist. A lot of time, young generation, they lose their patience. They think it is taking too long to let me get to that position. Okay. There's no royal road for learning, for being a scientist, for being everything you want to be. Okay. There's no royal road. Important vision, okay? The future is counting on you, okay? I know many people here are doing stem cell research. What's your vision? What's your perspective? Can you see someone like this, okay? A change in stem cell research could help these parents. Okay? Truly, the future is counting on us, on you. Okay? You will do something to make that happen. Do something to make that change. Okay? Future is counting on you. And stuff is very important. I really encourage you to think deep, think big, to have a big dream. I want to be a scientist. Another important vision. Okay? You're doing science. Okay? Because you want to use this science to serve humanity rather than the other way. Okay? Don't let humanity be enslaved by the science. The reason I think that's important because you see a lot of scientists at the end, they make a big mistake. And it totally changes the picture. And here, give me a cartoon here. A lot of scientists, they use their extreme means to get everything published because it was such a tough road. So sometimes they fake data. In the end, what? All these papers published full of fake data. You lost your humanity there. You want to be a scientist, you want to seek the truth, right? You want to do the best to find out the most important things a human being never explored before. If you fake data, you cross out everything. Okay? Don't do that. Don't let your humanity be enslaved by science. Science is tough. Be tough as it can be. And you have to stick it out. Okay? But don't do one thing. You need to stick out the principle. Okay? People faking data for certain reasons. For promotion, for business reason, for other purpose. And it's not worth it. Because in the end, what you do it has become part of human history. Everybody will find out whether you fake it or not in the end. Keep that in mind. Okay? Don't let science enslave you. It's very important. Faking data is just one thing. Okay? Scientists could be a very nice, gentle people. Sometimes scientists, you see them, they are square, they're kind of weird, they are weirdo, you know. It's not the way it should be. You could be like a normal person, you could be still honest, you could be still be dedicated, enthusiastic. Okay? Need not to be a strange scientist. Eccentric. Okay? There's an English term called eccentric. A lot of the previous comic book or whatever always describe scientists as a kind of weirdo, okay? Eccentric person. But that's not need to be true. doing research for humanity. Okay? Many of you, I don't know, many of you do animal experiments. Raise your hand. Anybody doing animal experiments? Nobody? Okay, see, one or two. I encourage people in my lab doing animal experiments. Do it responsibly, because there's a life involved. Okay, sometimes we do an animal. Mice, we need to sacrifice when we finish the experiment. Think about humanity. Okay? These mice dedicate their life for our research. Okay? So when you want to sacrifice, if you could do it, you have religion, say prayer for them. Show your sincerity. If you don't have any religion, just say a simple thank you will be okay. Because you do it responsibly. Okay? And tell people in the lab, in my lab, that's at least something you should do. When you sacrifice them, they thank you. Thank you for helping me doing my research. Okay? So that's to show your humanity there. It's very important. Okay? Don't lose that. Okay? Humanity is so important. Okay? You being a scientist, we want to dedicate these scientific effort for our humanity, for the benefit of our human knowledge, to be able to unravel the mystery, be able to do something good to contribute back to our society. We come up with a vaccine, come up with a certain novel strategy for cancer therapy. We're doing something back to return out to contribute to our humanity. So don't lose that. But when you are in a good situation or in a bad situation, there's always a challenge, always. And that's what makes research alive. Here the cartoon shows. I could see the stem cell lab. Okay? Maybe it's kind of in a situation, it was so crowded, there's no air conditioning, say. It's very, very much more difficult situation. You need to appreciate, you have the opportunity to do things there. Always show your appreciation. Because in the end, maybe you go to a luxurious lab space and you see all the beautiful bench, you see the beautiful equipment there, but nobody's doing experiment there. Nobody cares about doing experiment. What is the good of it? No good, still no good. Okay? So there's always challenge there. So the best situation, if you go to such a nice setup there, do appreciate that possibility to do experiment. Because there's always deficiency. We in the MD Anderson Cancer Center, our last setup was an okay situation. But we try to do our best to get our research done. Some of the luxurious lab, we call it 6-star lab. Sometimes you go there, you don't see that many people doing experiments. What is good about it? Appreciate every situation you have. This is always a challenge. Okay, so the last slide I want to share with you. We for the past 30, 40 years, our research in science, in biological science, advanced so much. In the old days, we could just do, my postdoc training, when I sequence a gene, we could publish a good paper. And right now, you don't do sequencing anymore. Okay? In the old days, a very good scientist, a biochemist, if they know any one of the oncogene here, that's already important discovery. That is the old days. Right now, with the new advanced technology, you can grab so many information at the same time. Think Lin or Dr. Yang or intelligent. Some of the important bioinformatics, you could dig it out quicker. But you still need to do analysis. And I would think that the technology will bring us to the next stage. And it's all coming on you. Okay? You are the newest generation of scientists. who will achieve a lot, and we're counting on you. Okay? Our hope is on you. Okay? Okay, you want to stop here? Come on. Okay. Thank you. Any questions? Any questions? Any questions? No?
Thank you for your talk. It's very interesting. I have a question I want to ask why. So when I finish the writer school and become a PI, very young PI, I find it very difficult to be a good PI. So could you give me some advice?
To be a good PI. Sure. I mean, it continues to have that characteristic of humanity is very important part. I think that you should be also still be caring, be patient, be able to guide students. And I think the important thing is